The discovery of recently identified genes associated with depression holds the potential to pave the way for innovative treatment approaches. Greetings and salutations to everyone. Today's video focuses on a groundbreaking study conducted by UCL researchers, which has identified over 200 genes linked to depression. Published in Nature Genetics, this research marks the first large-scale global study of the genetics of major depression, encompassing participants from diverse ancestry groups. The study has unveiled more than 50 new genetic loci and 205 previously unknown genes associated with depression. Notably, the findings suggest potential for drug repurposing, as one of the identified genes codes for a protein targeted by a widely used diabetes medication. Additionally, the study highlights new avenues for developing drugs to effectively treat depression. Without further delay, let's dive into our discussion. In the groundbreaking study published in Nature Genetics, researchers discovered over 50 previously unidentified genetic loci, which are specific positions on chromosomes, as well as 205 novel genes linked to depression. This comprehensive global study represents the first of its kind to explore the genetics of major depression in a diverse range of ancestral groups. In addition, the study highlights the possibility of repurposing drugs by identifying a gene that codes for a protein targeted by a widely used diabetes medication. Furthermore, it identifies novel targets for potential drug development in the treatment of depression. The prevalence of depression is widespread, yet there remains a lack of understanding regarding its development. Utilizing extensive genetic data, researchers have opened up new pathways to comprehend this illness and have identified numerous genes linked to depression. Each individual gene only contributes a slight elevation in risk. Additionally, this research has the potential to identify fresh targets for medication. However, investigations have primarily concentrated on individuals of European descent, which the researchers highlight as a significant limitation, particularly when studying a condition as intricate as depression. The international research team conducted a comprehensive study that utilized various genetic research techniques, such as genome-wide association studies, a meta-analysis of existing data, and a transcriptome-wide association study. They analyzed genetic information from 21 different study cohorts, consisting of individuals of African, East Asian, South Asian, and Hispanic Latin American descent. The study included a substantial sample size of nearly 1 million participants, with 88,316 individuals diagnosed with major depression. Significant progress has been made in the study's identification of genes associated with the risk of depression. This includes the discovery of new links and the reinforcement of existing evidence. Notably, the gene NDUFAF3 has shown potential implications for the development of drugs. The protein encoded by NDUFAF3 has previously been linked to mood instability and is targeted by metformin, a commonly prescribed medication for type 2 diabetes. Animal studies have indicated a potential connection between metformin and reduced depression and anxiety. Therefore, this latest finding emphasizes the need for further investigation into the relationship between metformin and depression. The study has identified additional genes that may have plausible biological connections to depression, including a gene associated with a neurotransmitter involved in goal-directed behavior, as well as genes responsible for encoding a specific type of protein that has previously been linked to various neurological conditions. To the researchers' surprise, the degree of genetic overlap in depression among different ancestry groups was lower than anticipated, measuring at approximately 30% using a novel method devised by the research team to assess the transferability of genetic associations between different groups. This level of overlap is lower than what has been observed for other traits and illnesses in previous studies. Consequently, it becomes increasingly crucial to examine depression in diverse populations, as certain findings may be specific to particular ancestral backgrounds. According to Professor Caroline Kuchenbecker, the lead author from UCL Psychiatry and UCL Genetics Institute, it is evident that our comprehension of intricate illnesses like depression will remain inadequate unless we address the Eurocentric inclination in genetics research and explore diverse populations worldwide for potential causes. The impact of certain genes on depression risk may be limited to individuals of European descent, highlighting the importance of diverse genetic datasets to ensure that genetic research can lead to the development of inclusive drugs benefiting individuals from all ancestral backgrounds. The study was led by Professor Kuchenbecker, who was joined by Dr. Xiangrui Meng, PhD researcher Georgina Navali, and Dr. Olga Giannakopoulou. Additionally, the collaborative consortia involved in the study comprised the Psychiatric Genomics Consortium, Major Depressive Disorder Working Group, China Kadoorie Biobank Collaborative Group, the 23andMe Research Team, Genes and Health Research Team, and Biobank Japan project. Professor Kuchenbecker emphasized that the current discovery effort is only the beginning, and further research will be necessary to validate these newfound targets. However, the initial identification of these targets has proven to be a significant and crucial undertaking, particularly considering the urgent demand for new medications in treating this disorder. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.